The other kind of movement is microtubule based, and this is for the cilia and the flagella in the cell. The cilia typically are shorter than flagella. You can see here that the surface of this lung cell has many, many, many cilia on the surfaces, and they often sweep liquid over the surface, or they can propel the cell through the liquid. Flagella are longer and typically are used to propel the cell forward and have a wave-like beat. Okay, instead of actin, these use microtubules. Microtubules are these hollow tubes that have 13 protofilaments, so we use 13 of these to make up our hollow tube. Each of these protofilaments is made up of a dimer of alpha and beta tubulin. We also have a plus end and a minus end. So the plus end is where we add and lose filaments from, in this case. So when we're growing, we're going to be growing towards the plus end. These have motor proteins that walk on the microtubules, kinesins and dyneins. They each have a direction that they move. And dyneins are particularly important for flagellar and ciliary-based movement because they are what is going to generate the waveform of the flagella or the cilia by walking along the neighboring microtubule. We'll look at that in a second. So if we look at the way these are set up in a flagella or a cilia, this is a cross-section through one, and we can see a 9+2 microtubule array of these microtubules. And what that means is we have 9 outer doublets, and this is 2 microtubules attached to each other. And then in the inside, we have 2 central microtubules, single ones. So we have 9 outer doublets, 2 central microtubules, and then there's accessory proteins that help set up the movement. The first of these is the outer and inner dynein arms. So these are the motor proteins that do the actual movement. So the tail is attached to one microtubule, the head group walks along the neighboring microtubule. Then we also have these things called radial spokes, and a radial spoke is controlling the movement. So this is like the control center. It is determining when the flagella or cilia is beating, when it's not, what part of the cilia or flagella is moving. And then finally we have this nexon, and this is a connector. And this basically holds together the neighboring microtubules. And if we look without the nexon and we add ATP, these two slide apart from each other. If we add these linking proteins, the nexon, then those microtubules are held together, and then we get a bending motion instead of a sliding motion. And this sets up the waveform of the flagella or the cilia. Okay, what we're going to do in lab is you will get to actually look at an organism that has flagella. The Chlamydomonas reinhardti is a freshwater algae. It's single-celled, and it has 2 flagella here, the anterior end. And it swims with these 2 flagella. It's also photosynthetic, so it'll appear green. And there are many interesting mutations that already exist, and you will get to look at a few of these mutations and look at their altered swimming pattern when they have mutations in the flagellar proteins.